everyone, welcome to exam 4, my second essay video for exam 4. Today we're going to be talking about our lungs and inhalation, expiration, gas exchange, and how it all works. So first things first, ventilation, breathing in and out, is all about pressure. Where is our gradient, our airflow going to? So we should be pretty familiar with the fact that air, just like everything else in nature, wants to go from high to low pressure, right? That's another way of saying from a state of high energy to a state of lower energy. And this is true for ventilation. So when we're breathing in, your body is doing things to create a negative pressure inside of your chest, inside of your lungs, which is what takes in the air from outside in the atmosphere and brings it into your lungs so that you're able to breathe. So let's go through some simple anatomy real quick. We have a person who is breathing in, and inside of that person they have a set of lungs. So this is my really bad drawing of lungs connected through the bronchi, going up into the head. So we have two major muscles that help us with our breathing. The first one is going to be a dome-shaped muscle in the bottom beneath our lungs. That's called our diaphragm. The second one are a bunch of these smaller muscles that are in between our ribs on the side of our body, or on the side of our lungs. Those are called our outer intercostal muscles. When we're breathing in, our diaphragm contracts. In its relaxed state, the diaphragm is in a dome shape like this. When it contracts, it's actually pulling down. So pulling down, that creates a greater amount of space in our thoracic cavity. And that greater amount of space, or that greater volume, creates a more negative pressure, because pressure and volume are inversely related, right? We have, if our pressure, or if our volume goes down, then our pressure will go. If volume goes down, then pressure goes up, and vice versa. If volume goes up, then pressure goes down. And so if we're increasing the volume, we have a lower amount of pressure. It becomes more negative inside of here when compared to outside in the atmosphere. And so because we have that difference in pressure, we have a gradient. So that air will come in through our nose and down into our bronchi and then into our lungs. That's like the very basics. Now there's something called a pneumothorax. So this is what happens when we have a something that messes with this ability. So if we get a broken rib that can maybe like puncture our lung, then we have a difference in pressure inside of our lungs as well when opposed compared to our interstitial space here, or pneumothoracic space. So when this happens, we can get if we get a punctured lung from like a broken rib or even a stab wound, we can have our lungs collapse and they'll get a lot smaller, and as they do so, we're unable to really use that lung, and then we can get pressure buildup inside of the space that's in between, the rest of the body and the lung. And in order to treat this, you need to make an incision so that you can allow the building pressure that's inside of the pneumothorax to escape, and that allows the lungs, there's less pressure crushing it, and so it'll allow it to come back out to its normal size. there's a couple of factors when we're talking about lungs and gas exchange. What we're really talking about is the smallest anatomical portion of the lung, which is the alveoli or alveolus. Alveolus, you have your bronchi going into your lung, right? And then inside of there you have your branches. And then at the very end of these, you have more tiny branches. And they kind of end in what looks like these clusters of grapes. They kind of look like grape clusters at the end. If we zoom into one of these, this is what we call an alveoli. And basically they become sacs of air. So when we're breathing in, we're taking all the gas that we're breathing in and we're putting it into the clusters at the end of these branches here. And when the gas comes in, it has to get through six spaces in order to get from the alveoli space in here and into a capillary to attach to blood. And those six spaces are including, they involve the fluid covering the inside of the alveoli. They involve what's known as squamous cells that line the alveoli. Squamous means like thin or scale-like, and the thinness of those cells is what allows air to diffuse easily past them. And it has to go through the basement membrane of the alveoli, and then there's a tiny space between the blood vessel and the alveoli. It has to go past that, and then past the basement membrane of the capillary, and then through the squamous cells of the capillary again. The scale-like cells are present in both alveoli and our capillaries inside of our lungs. There's something called the law of Laplace. This will help us understand something really important to know about alveoli. So the law of Laplace, I'm just going to write his name, Laplace, came up with a law where P is equal to 2T over R. P is equal to pressure. In this instance, we're going to take pressure and treat it like surface tension. So in our lungs, we're breathing in, we have a lot of humidity inside of our lungs. And that humidity is so high that we actually have kind of like a watery film over all of our alveoli. And this water molecules are attracted to one another, and so they kind of want to grab onto each other. If you've ever seen two beads of water touch, you'll notice that they kind of hold onto each other and they become one bigger bead. Well, this is what happens on the surface of our alveoli. We have this water that kind of lines or makes a film around this. And because water wants to hold on to itself, it creates tension or pressure in this case. And that pressure wants to make that alveoli as small as possible so that the water molecules can get closer together, if that makes sense. Now, how we calculate this pressure, or another way we say it is surface tension, is we take 2 times T, which is tension, and put it over R, R being our radius. So what does this tell us? Well, if we have a smaller radius in our alveoli, if our alveoli are really, really small, it's going to be a lot higher pressure. Because if we have, like a 1 down here and a 5 up here, 5 times 2 equals 10, 10 over 1 is equal to 10. That's a lot bigger than, say, if instead of T being 5, say T was 0.5. 0.5 times 2 would be just equal to 1. 1 over 1 is equal to 1. And so you see this makes a really big difference depending on how big well, that's with T. If it were R, instead of changing our T, if we took 2 times 5 is equal to 10, and then say our radius was now, instead of 1, say it's 2, instead of being a total surface tension of 10, we would have a total surface tension of 5. So essentially, the larger the radius of our alveoli, the easier it is to expand them and to breathe in. Now, there's another factor to know that's important to note is we have this thing called surfactant. Surfactant is this substance that has both amphiphilic, that has an amphiphilic effect, which means it is both water-loving and water-hating. So basically what it does is it decreases the surface tension. It disrupts those bonds between between the water molecules, and it decreases that surface tension. So water isn't holding on to itself as much. And so surfactant will make it easier for us to breathe in. And so a lot of times when newborns are first born, they have kind of underdeveloped surfactant. So they don't have all the normal amounts of surfactant that we do, and that can end up causing a problem if they don't get that developed quickly, because the less surfactant that they have, the more surface tension they'll be experiencing in their alveoli, and the harder it will be to have them breathe in. Now let's talk about our alveolar gas equation. So, alveolar gas equation, this is something that's a little tricky, but we'll get through it. I'm going to write it out. My whiteboard isn't going to be big enough for just one line. I'm going to have to break it up. Okay, so basically our alveolar gas equation solves for our alveolar O2 pressure. We take our FiO2, which FiO2 is the amount of oxygen that is present, I believe. Let me double check that that's true. It's the fractional concentration of inspired oxygen. So usually FiO2, it's just the percentage of air that you breathe in that is actually oxygen. Our atmosphere is not pure oxygen. If it were, our physiology would be way different, or we wouldn't be able to survive on the planet. But since oxygen in the atmosphere is always hanging out at around 21%, which means that this number is usually 0.21. 0.21, 21%. You multiply that by the pressure in the atmosphere, subtracted by the pressure of the water vapor inside of our body, which is usually about 47 millimeters of mercury. And then you subtract it by this ratio here, which is our PaCO2, so the pressure in our alveoli of our CO2. PaCO2 normally is about 40 millimeters of mercury. And then our R is this ratio that is equal to the volume of your CO2 versus over the volume of your O2. This ratio here is due to the interactions of oxygen and carbon dioxide, and it kind of makes an assumption that inspired air contains no carbon dioxide, but we know that's not true. But for the time being, we just kind of ignore that until Unless you live in a place with really poor air quality, then I guess you would have to take that into account. For our purposes, we will not be taking that into account. So, what can this equation tell us? Well, this equation basically tells us how much oxygen, fresh oxygen, we're getting with each breath. So ideally, we want this number, our PaO2, to be around 0.85. If we can have our proper levels of oxygen, then our inspiration is doing its job. We're getting enough oxygen to get our blood oxygenated, which then can oxygenate all of the tissues that needs that oxygen, primarily the brain, and then the heart, and then so forth. So CO2 and O2 are transferred in the blood through red blood cells, mostly. We have carbon dioxide as well as oxygen that attaches onto our red blood cells. And our red blood cells have these things called hemes, or hemoglobins. And this is what makes our red blood cells so effective at transporting these important gases into and out of our body. Hemoglobins will attach to O2, oxygen, as well as carbon dioxide. And even though that they're kind of the main source of our body's oxygen, oxygen cannot go straight from a hemoglobin on a red blood cell into a cell nearby. It has to first diffuse or be dissolved into the blood, and from there it will go into cells that need that oxygen. And how can you tell if a cell needs oxygen? Well, if there's a gradient, right? So our red blood cells contain the majority, about 98%, of our oxygen in our body, while the blood itself, like the plasma, contains only about 2% of our oxygen in its dissolved state. It's important to note that red blood cells do not directly oxygenate cells. Rather, it's the oxygen that diffuses off of the hemoglobin and then into the cells that need it. When it comes to carbon dioxide, we have carbon dioxide can enter our red blood cells, our CO2, which then can combine with water and create our favorite base, which is bicarbonate. And 70% of our carbon dioxide is actually in the form of bicarbonate in our body, which is why it's okay to not get rid of all of our CO2 with every single breath that we have, because we actually use bicarbonate a lot, and it's a very useful base that keeps our pH levels where they need to be. All right. Oxygen and hemoglobin dissociation curves and factors that influence affinity of hemoglobin. So, when it comes to clinical applications of oxygen levels inside of our blood and carbon dioxide levels inside of our blood, we have this really useful way of measuring this called the oxygen hemoglobin dissociation curve. That's kind of a mouthful, so let's break it down. So here I drew the graph. So on the y-axis we have oxygen percentage of saturation of a hemoglobin, so the amount of hemoglobin that is saturated with oxygen. Then on the x-axis we have our pressure of oxygen in the blood, so how much oxygen is in the blood. And we don't really want to worry about units right now, we just want to get the concept down. So this is what a normal dissociation curve looks like on this graph. If we have a line, if we shift our normal S-shaped line to the left, then we have our lower CO2 if we're shifting to the left with higher oxygen, which makes sense, right? Because shifting to the left, we're not needing as much pressure to get that percentage of saturation higher, right? It takes less pressure, less work to get that hemoglobin really to saturate with our oxygen. It also indicates a decrease of temperature, so this can happen if you're experiencing hyperthermia, hypothermia, and it can also be due to alkalosis. Now remember, alkalosis, if we have respiratory alkalosis, our CO2 is down, which makes sense because a signal of a left shift is having the CO2 down. Now, let's say we shift this graph to the right. What does that mean? Well, shifting our dissociation curve to the right is the exact opposite of shifting it to the left. If we shift it to the right, then our carbon dioxide will go up, which is an indication of respiratory acidosis, and our oxygen levels will go down, which you can see here because it takes more pressure. As we're going along here, it takes more pressure to get the same amount of percentage of saturation within our hemoglobins. So we're having to do more work in order to get the same amount of oxygen that we need. And it also indicates a higher temperature. So if you're experiencing with heat stroke, this could happen. And like we already mentioned, it is a cause of acidosis. Well, thank you for watching my video. Hope you guys have a wonderful rest of your week. See you next week.